Hi everyone, my name is Naomi Codrington. The title of my research presentation is called Genetic Mosaic Analysis of the Circadian Control of Sleep and the Drosophila Brain. Circadian clocks are a natural internal process that regulates the sleep-wake cycle in a temporal pattern and repeats roughly every 24 hours. These clocks consist of transcriptional feedback loops and are highly conserved genetically. In picture A, it shows that at the molecular level, the period length of circadian rhythms is determined by the period gene. So to your right, in picture A, this is the period gene. Expression of the clock genes, period or per, and timeless, or TIM, is activated by transcriptional factors clock and psych. So right here in picture A, this is TIM, this is clock, and this is psych, which is transcriptional factors. Mutations in the per gene can shorten, extend, and even inhibit the period of the circadian rhythm. In most animals, sleep is controlled by both a homeostatic control system, which promotes sleep after prolonged wakefulness, and a circadian clock, which is thought to set thresholds for sleep and wakefulness. In graph B, you can see that there's an increase in sleep pressure the longer you stay awake until you reach the high threshold. So here in graph B, this right here will be the increase in sleep pressure, and then this right here is the threshold, the high threshold. When you fall asleep, the sleep pressure decreases until you reach the lower threshold, and then you wake up. So here, the sleep pressure decrease, and this is the lower threshold. This happens in a temporal pattern, and the threshold is set by the circadian clocks. The daily changes in the upper and lower threshold set sleep timing. In the fly Drosophila, the molecular circadian clock is expressed by only 150 neurons. This network is responsible for the production of an endogenous circadian rhythm, and some anatomical classes of clock neurons within this network are thought to promote sleep or wakefulness. In the picture above, the neurons in blue are wake-promoting neurons. The ones here to the left, the ones here to the right, and the ones here above. Though the circadian timekeeping functions of the various classes of clock neurons has been well studied, the role of neural timekeeping in the control of sleep timing has not been examined. Here I have focused on the role of circadian timekeeping within the wake-promoting large ventrolateral neurons and the control of sleep, which are these neurons. In my experiment, I used the split Gauss system to manipulate the molecular clock in a specific subset of clock neurons in the fly brain. In this system, two Drosophila enhancer sequences drive the expression of two halves of the Yi transcriptional factor Gauss4. In the picture to the right, the first half has the binding domain, and the second half has the activation domain. Gauss4 is therefore reconstituted only in cells in which both enhancers are active. In these cells, reconstituted Gauss4 promotes the expression of the transgene flow downstream of the upstream activating sequence, or UAS, from yeast, which is also shown here in the picture to the right. In my experiment, I used a split Gauss4 system to express a dominant negative form of the clock protein delta psych, which disrupts the translational feedback loop in cells that express it. Versions of the split Gauss4 driver line GMR 5500645 to UAS delta psych male flies to disable circadian clocks, specifically in the wake-promoting large ventrolateral neurons. Each of the parental lines was crossed to a genetic backbone strain to produce control lines. Daily patterns of sleep were recorded using the trikinetics Drosophila activity monitoring system, which can hold up to 32 flies. Beam crossing were collected in 1-minute windows, and sleep, which has a minimum requirement of 5 minutes of inactivity, was computed over 30-minute intervals throughout the durations of the experiment. From this, total minutes of sleep and average sleep bout duration during the day and night were computed using phase. Statistical comparison between genotypes were done using a Kruskal wireless test followed by a pairwise Wilcoxon test for post-hoc comparisons. These analyses were performed using custom scripts on R. So here's my results. According to the graph, the experimental flies, which contain both GAL4 and UAS delta site, displayed much lower levels of nighttime sleep and a modest reduction in daytime sleep compared to flies that only had GAL4 and flies that only had UAS delta site, which are my controls. Also in the graph to the left, there was a significant reduction in total sleep and nighttime sleep. In the graph to the right, it shows that reduced sleep in the experimental flies was driven by a decrease in the duration of sleep bouts at night. In conclusion, when the molecular clock is suppressed specifically in the large ventricular neurons, it leads to a significant reduction in total sleep, driven largely by a reduction in duration of nighttime sleep bouts, and expected instead for an increase in sleep at the beginning of the day. This result suggests that nighttime sleep is normally promoted by large ventricular neurons, so a re-evaluation of the roles of clock neurons in sleep control is needed. I would like to thank the CSURP program, Dr. Schaffer, and the members of his lab, Avalash, Imani, and the rest of the staff members for my summer experience.